Welcome to our 17th video as part of your IB Biology course. This video we are looking at cell respiration, and that is unit 2.8 of topic 2, molecular biology. Okay, first of all, let's clarify a couple of pieces of information. First of all, respiration is not breathing. Respiration is a biochemical process that converts the chemical energy in glucose into chemical energy in the form of ATP. We'll get into that further. Ventilation, the physical act of inhaling and exhaling, which you guys could call breathing, is only part of this whole process. This is how the oxygen gets into the lungs and how the waste products such as carbon dioxide are exhaled. Gaseous exchange is just part of the transport of the gases required for respiration. Okay, so this sums that up. We have breathing on the left-hand side. As you can see, oxygen being breathed into the lungs, carbon dioxide is being breathed out. Inhale, exhale, inhale, exhale. However, that oxygen that has been breathed in then has to travel to the mitochondria for cellular respiration to occur. And one of the waste products of respiration is carbon dioxide, which then travels from the mitochondria out to the lungs and is exhaled. All living things need energy. What type of energy is needed though? So we need this stored chemical energy, and we get that from sugar. Okay, that sugar, that stored chemical energy, is produced during photosynthesis. So the sunlight is fixed into that sugar. Now this is released or made usable through the process involving ATP, which we'll look at shortly. And this all occurs through cellular respiration. So here we have a beautiful infographic showing what I'm talking about. That light energy is fixed in the chloroplast during photosynthesis. We look more into photosynthesis in our next video. That is fixed into the molecule or into a sugar molecule, glucose fructose. And along with that sugar molecule plus oxygen in the mitochondria, ATP is produced, and carbon dioxide and water are released as products of aerobic respiration. We'll also be looking at another respiration, anaerobic respiration, but that's the nuts and bolts of it all there. Okay, so time to introduce ATP and ADP. All right, ATP, adenosine triphosphate. Let's break this down. Triphosphate, phosphate, phosphate, phosphate. Okay, so we see we've got hydroxide there. We've lost hydrogens along the way through, what do you think? Okay, condensation reactions form water, and hydrolysis take water in. So I reckon we may have had some condensation reactions to form our ATP. Okay, so that's ATP. Here we've got ADP, diphosphate, adenosine diphosphate, 2 phosphates. What's the difference between ATP and ADP? ADP has 2 phosphates. ATP has 3 phosphates. Okay, ATP is a source of energy. ATP from cellular respiration is immediately available as a source of energy within the cell. Cells require energy for three main types of activity: synthesizing large molecules like DNA, RNA, and proteins. So synthesizing meaning making, okay, so making large molecules. Pumping molecules or ions across membranes by active transport. And moving things around in the cell, such as chromosomes, vesicles, or in muscle cells, the protein fibers that cause muscle contraction. That's pretty cool because we've now studied these things, so that should actually all make sense to you. This one maybe not quite so much about the muscle contraction, but overall that should all make sense. Energy is required for those things, and the form of energy that the cell uses is ATP. Okay, energy in the form of ATP, adenosine triphosphate, is the energy that drives all metabolic processes within the cell. Energy is released for use by the cell when ATP is hydrolyzed, so broken down, okay, as follows to produce ADP. So ATP loses a phosphate to become ADP through a hydrolysis reaction. And also released there is an inorganic phosphate. Okay, so we've got respiration, and during respiration, ADP is converted to ATP. ATP is what your cells then use as energy. Okay, so we've got this process of glucose reacting with oxygen. Okay, and those high energy bonds there, when they're broken, that energy is then used to top up the ADP to ATP. All right, and that's a hydrolysis reaction. And then we form carbon dioxide and water. Sorry, that's not hydrolysis, I'm talking madness. All right, so ADP, you got ADP has less energy than ATP. We have to join the phosphate to the ADP to make ATP, and that's a fully charged battery ready for the cell to use. Alrighty, let's have a bit more of a look at the process of respiration. There are two main types of cellular respiration. There's anaerobic, which involves just a partial breakdown of glucose in the cytosol, and we only get a relatively small yield of ATP through anaerobic respiration. And then we've got aerobic respiration that utilizes oxygen to completely break down the glucose in the mitochondria. And for this, we get a larger yield of ATP. Just remembering, during respiration, our ADP is reacted to form our ATP. So for one reaction of respiration, you get about 38 ATP molecules. It's pretty impressive. Okay, anaerobic respiration. Let's have a look. So anaerobic respiration is not as efficient as aerobic respiration. Okay, there's a relatively small yield of ATP that comes from glucose during anaerobic respiration. Anaerobic respiration is useful in cells when there's a short supply of oxygen or when oxygen is not available at all. And we get these short but rapid bursts of ATP production when needed. Now, when we have a look at our anaerobic respiration, we're going from glucose to lactate. Okay, so remembering our sugars, glucose to lactate, again, ADP forms our ATP. This is occurring obviously in humans. Now, we know that lactic acid in our system causes muscle cramps and stitch and things like that. Okay, now in yeasts and plants, our glucose is broken down to ethanol and carbon dioxide during anaerobic respiration. So we've got our ADP to ATP still occurring, of course, because it is a form of respiration. But interestingly, that glucose is broken down to ethanol. Ethanol is alcohol, so we're going to see some uses of anaerobic respiration shortly. Okay, now looking at aerobic respiration. Okay, now we know that in our aerobic respiration, we've got that glucose reacting with oxygen. And due to those high energy bonds in the glucose molecule, that allows that ATP to form, that ADP to form ATP. And our waste products from the reaction are carbon dioxide and water. Now, aerobic respiration in cells requires oxygen, and it gives us a lovely large amount of ATP from the glucose. There are a series of steps involved in these chemical reactions. You need to be aware of each process. However, you don't need to know them in a huge amount of depth at this time. So first of all, we've got glycolysis. Now, this occurs in the cytoplasm. We have what we call the Krebs cycle, and with oxygen presence in a matrix in the inner compartment of the mitochondria, this is where the Krebs cycle occurs. We'll look at these a bit further in some diagrams in a moment. And then we have the electron transport or the oxidative phosphorylation. With oxygen present on the cristae, the inner membrane of the mitochondria. Again, we'll have a look where these all take place. Hopefully I don't have to say phosphorylation too much. Okay, so as I said, we've got glycolysis occurring in the cytosol here. Okay, that Krebs cycle in the mitochondria and the electron transport train, or that oxidative phosphorylation, occurring on the cristae in the mitochondria. Okay, another diagram here just showing that again. So you need to know where they occur rather than what they're doing. Okay, some applications and skills to do with respiration. So first of all, applications of anaerobic respiration. Yeast respires anaerobically to produce ethanol and carbon dioxide. So when you're fermenting raw ingredients, we can make beer and wine. That's really embracing the ethanol. And you can also make bread, okay? And that's where we utilize the carbon dioxide and trap it in the dough. The CO2 produced by the yeast is used to make bread rise. I believe I just said that. And while the ethanol evaporates away, so we don't end up with highly alcoholic bread, although you actually can. And bread without yeast is a very, very flat bread. Alrighty, so as I said, uses of yeast. You can make beer, you can make wine, you can make bread and other doughy products. Some other applications of anaerobic respiration you need to be aware of. During strenuous exercise, our muscles may use up all the available oxygen. Without oxygen, the muscle cells will then start to respire anaerobically and are able to produce a small amount of ATP, which allows for the power of the muscle contractions to be maximized. During anaerobic respiration in animals, that produces lactate, and we know that as lactic acid. So that's that muscle burn we can feel. Okay, so this is showing that anaerobic respiration in muscles, or it's showing us aerobic respiration and what happens when it's anaerobic respiration. Okay, so one of the things you need to know about in the application section is the use of respirometers. A respirometer is a simple piece of apparatus that can measure the amount of respiration. Cellular respiration uses oxygen and produces carbon dioxide and water, so we know that. We can measure the consumption of oxygen as an indicator of a respiration rate. So here we've got a respirometer. Oh dear, I'm having trouble with pronunciation today. In tube A, over on the left, the organism to be tested, or it could be a germinating seed, is positioned and the tap is closed. So we've got a little insect in there and we're testing the respiration rate of our little insect. Looks like a fly. The organism starts respiring, well, would actually continue respiring because it was respiring before it went into the container. So it's consuming oxygen and it's producing carbon dioxide and water. The alkaline solution at the bottom of tube A will absorb the carbon dioxide. So we've got that potassium hydroxide at the base of tube A there. That's going to absorb the CO2. Now, tube B is the control where no oxygen is being used and no carbon dioxide is being produced because there's no living organism present in there. The capillary connecting the two tubes is a manometer. The reduction in the oxygen in tube A will reduce the pressure in tube A. This will move the coloured liquid in the manometer in the direction of tube A. So it kind of gets sucked up because the pressure in tube A is being reduced due to the oxygen being utilised. So this provides an indirect measurement of oxygen being consumed, therefore allowing the rate, the amount of oxygen consumed per unit, to be calculated. Now, in terms of your nature of science, we need to have a think of some ethics here. Respiring organism. If using invertebrates rather than seeds, what are the ethical questions that need answering? Is it acceptable to remove animals from their habitat for use in an experiment? Can the animals be safely returned to their habitat? Will the animals suffer pain or any other harm during the experiment? And can the risk of accidents that cause pain or suffering to animals be minimized during the experiment? In particular, in this experiment, can there be any contact with the alkali? How can that be prevented? Is the use of animals in the experiment essential? Or are there alternative methods that avoids using animals? So you could use seeds or something. Thank you for watching this video, and I will see you soon in our next video on photosynthesis.